Hello nerds, welcome to Ahmad Coaching. Today we are going to study about DNA fingerprinting. DNA fingerprinting is a method which is used to determine the probable identity of a person based on the nucleotide sequence of certain regions of human DNA that are unique to the individual. So what actually is happening that we are using DNA fingerprinting and we are actually focusing on the certain regions of that DNA, as the name indicate that we are talking about the DNA. So we will just focus on the certain regions of the DNA which are unique to individual. The DNA fingerprinting is just like the fingerprints of each person. Similarly, the DNA fingerprints, they are also unique. So normally what we use fingerprints to identify the individuals, but in case of DNA fingerprinting we use DNA to identify the individuals. This DNA fingerprinting technique is widely used in a variety of situations such as criminal investigations, forensic purposes, and paternity test. The paternity test is actually a test in which you have to find out who is the parent of a baby. So in these situations, one aims to match two DNA fingerprints with one another, such as a DNA sample from a known person and one from an unknown person. For example, a crime happens somewhere, so the police collected the samples from the crime scene, and after that they performed the DNA fingerprinting. So they got this. Now they have some suspects. So what they did, that they also run the fingerprinting of 3 suspects and then match the DNA fingerprinting of the fingerprint they collected from the crime sample. So what you can see, that these are the unique markers or unique fingerprints. Now they are going to match these unique fingerprints with the suspect. So we can see here this one and these two green bands. We can see that the suspect one doesn't have the band at the exact position, but the suspect 2 has. Similarly, the suspect 3 doesn't have at the same location. Then comes locus 2 or the second band. So you can see that suspect 1 doesn't have at that point, but suspect 2 has these 2 bands at the same location, even though suspect 3 also has the same bands. Then comes the third one. The suspect 1 doesn't have, the suspect 2 has, while the suspect 3 don't have. So we can see that the fingerprints 100% matches with the suspect 2. It means that crime scene samples, they are from suspect 2. Now the question is how they are getting these bands, or how you are going to perform the DNA fingerprinting. So there are the steps of DNA fingerprinting. First of all, you are going to take the samples. For example, we are studying some crime scene, so we can collect the blood samples from the crime scene, and from the blood sample we are going to extract our DNA. So we have the DNA. The next step is that we are going to cut this DNA into small fragments by the help of restriction enzymes. So we have, you can see that we have excised, we have cut all the DNA. After that, we are going to separate the DNA fragments by the help of electrophoresis on the agarose gel. The agarose gel is actually like this. It is a gel which has some pore size, and the sample of DNA, it runs from top to bottom, and they are going to form the bands according to their size. The bigger one will stay at the top while the smaller one will be at the bottom. So you can see that from here to like this we run our samples. After that, the DNA band pattern in the gel is transferred to a nylon membrane by the help of southern blotting. So we are going to place a nylon membrane on the agarose gel and then transfer the bands from agarose gel to the nylon membrane. After that, we are going to add our radioactive DNA probe. on the nylon membrane, and these DNA probes, they are going to bind with the bands on the DNA sequence on the membrane. After that, we are going to remove the excessive DNA probes by the help of washing, and only DNA probes which are bound with the DNA will remain on the membrane. After that, we are going to place our X-ray film on this membrane, and then X-ray film is going to capture the radioactivity from the probes which are bind with these DNA samples, and then we will remove the X-ray film, and we can see that on the X-ray film we have visible bands, and these bands are known as DNA fingerprinting, just like this one. And from these bands we can find out whatever we wanted to find out. We can find out the criminal, we can use it for the paternity test, and we can also use it for another forensic investigations. So this is what we called DNA fingerprinting. If you have any question related to this topic, you can ask me in the comment section. I will see you in the next video.